Okay, good morning, Dr. Lanning. This is the morning and Wednesday, a little bit before my interview for UCLA. But today I'm going to talk about the paper that was assigned to me, which is the synthetic immunomodulation with a CRISPR super-repressor in vivo. This paper was published in Nature Cell Biology, and the main author's name was Farzana Maghadam, and I found out she's actually a graduate from the PhD program at Arizona State University, one of the schools I'm applying to. And in this paper, they talk about how they repurposed a certain CRISPR technology and gave it an enhancement, or they enhanced it in ways that modulate an immune response in cells by controlling both transcription and DNA for a specific immune-related gene. But before we get into the nitty-gritty of the paper, it's really important to understand some background around CRISPR, surrounding CRISPR, and how it enables biologists like myself and yourself how to regulate transcription at a very precise level. So CRISPR stands for clustered, regularly interspaced short palindromic repeats, and it was found in bacteria to be a defense mechanism against, in their genome, it was found to be like some sort of defense mechanism or immune response against viruses. And the attention to CRISPR technology has been recently advanced. As you can see here, there's been a recent spike in the amount of papers that use CRISPR, that are focusing around CRISPR. CRISPR wouldn't work without the Cas9 protein, and the Cas9 is a protein that was found in the bacteria, but for purposes of using it as a molecular tool, scientists has utilized it and taken advantage of its binding affinity to, having those nuclease domains, which is shown here as a scissor, RNA binding domains and DNA binding domains. And what the Cas9 does is recruit a guide RNA and bind to specific DNA that matches the guide RNA, and the Cas9 will use its nucleus domain to create a double stranded break, and these are its domain, their nucleus domains. But the question within all this function using CRISPR is can scientists repress transcription without exerting a permanent damage to the DNA? And this is sort of the background behind the Cas9 system. And what people have found out is using a dead Cas9, a catalytically dead Cas9, meaning a Cas9 with no nuclease domains, enable scientists to use a protein like Cas9, a guide RNA, to locate the DNA, the loci, but not cleave its DNA. And this is really important because it's effective for transcriptional repression or activation or some sort of epigenetic modification to it. Thirdly, it's also been found out that the length of the guide RNA serves as some sort of enhancer or allows the Cas9 protein to cleave DNA. And here, this was a paper published by Samara et al., and this was one of the papers that our paper referenced, and it was showing that the guide RNA length really allows the CRISPR-Cas9 to cause cleavages or splice the DNA, and it's recorded here in the percent of deletions in their samples. And you can see that there's a difference starting at 16 nucleotides. So the aim of this paper was if they could enhance repression by using some guide RNA that's less than 16 nucleotides with a dead Cas9 protein. So the study objectives, or the overall schematic, or overall the overview of the paper, was many things. And they all revolve around looking at this sort of model system or this technological system where they have a Cas9 protein that does not have nuclease activity, a guide RNA, a very, and we'll talk about it. I'll talk about it later in this talk. But a guide RNA that varies in nucleotide length, repressor domains that bind to the Cas9 protein, and there's a variety of repressor domains that they tested. Use these three in conjunction to target genes and block transcription without harming the DNA, and see if they could enhance or repress DNA at a more superior level compared to the previously published findings. And so that was the first aim, which was to evaluate various transcriptional repressors and domains in vivo. And then is to determine if the system can modulate. So not only can it repress the gene, but can we see some sort of response? Can we see an immune response in vivo and not in vitro? Thirdly is to determine the long-term effects of using CRISPR as a transcriptional repressor. This could be using this Cas9 system to repress the gene and see the effects long-term. In this case, it would be mice, but you would be the authors measure side effects like LDH levels, I think it was certain metabolite levels as well. And they, and lastly is, can this system be used to prevent disease or counteract a septicemia? And this is really important because they test whether this immune strategy or this CRISPR strategy can be a prophylactic, or if it can reduce the sepsis within a system before and after. And so overall, this paper uses this system, but first they needed to figure out the repressor domains, and that's what the results in figure 1 demonstrate. So here I'm showing you the first figure of the paper, figure 1B, I believe. Yeah, figure 1B. And yes, it's testing the effectiveness of various repressor proteins on RNA expression in vitro. So previous studies had found a variety of repressor domains, such as CRAB and CRAB MBD2B, and they tested the effectiveness of the repressor against the relative mRNA expression. And what they found was a combination of these specific repressor proteins, which is the MS2, HP1A, alpha, and CRAB. And I have the names of those repressors. I should have, oh yeah. It's a heterochromatin protein 1, alpha, and then a Croupen-associated box repressor domains. And as you can see, across all these genes that they repressed, that combination of repressor proteins significantly reduced the amount of mRNA expression when compared to the other combinations of repressor proteins. Next was to test that combination of repressor proteins and its effectiveness using truncated or elongated guide RNA length. So for moving forward, 14 nucleotides and a length in a guide RNA would be referred to as truncated, and 20 would be referred to as elongated. And so what they did in this design was cleave, or they transfected cells in vitro with either a 14 nucleotide or 20 nucleotide length guide RNA. And then they measured repression, transcriptional repression, and expression of these two genes, these two nucleotides. And this is what they found. So figure 1D demonstrates that using both, or there's no difference between the effectiveness of repression when you're using the 14 nucleotide or the 20 nucleotide guide RNA. But then you could ask, well, What if it's the delivery? What if there's less 14 versus 20 within these cells? And to answer that, they correlated the amount of elongated guide RNA versus truncated guide RNA. And what they found was that there's a strong correlation between these two the guide RNAs. Next is using the 14 nucleotide length against a specific gene. And that gene is also demonstrated here, but it's demonstrated here. And they wanted to see whether they can repress, right, if their combination, now they're really putting everything to the test, where they're looking at the combination of repressor domains with the 14 nucleotide length guide RNA and the repression of MYD88, which is a myeloid differentiation primary response gene, and it's known to turn on amongst when there's some sort of immune response in the cell. And what they're showing here is that using a truncated guide RNA is far more it's superior at repressing this gene, this transcription of this gene, compared to what was previously known here, which is shown in gray, which is the guide RNA for the MYD88 with not the combination of those repressor domains. But overall, this demonstrates that, yeah, our method is a stronger repressor and it doesn't harm the DNA. Next is to test the efficacy of that same, these tools, these CRISPR tools, which is using a combination of those repressors and the truncated guide RNA in vivo. And what they did for this is they injected mice, Cas9 expressing mice, with a vector, an adeno association virus vector that carried these guide RNAs. And they measured at day 0, and then they analyzed gene expression 21 days later. And so what they wanted to do was test whether repression of MY88 was significantly repressed across the lungs, the blood, and in the bone marrow. And what they found was, and their answer was yes, this system significantly represses this gene across various areas in vivo. And all the data is shown in pink here, and the expression is significantly lower to their controls, to their mocks, and to what was previously known to be the most enhanced repressor, which is shown in gray. And they also found this to be true for genes like ICAM1 and TRNF alpha, which are genes that are known to be known to interact with MYD88 downstream of its activation. Additionally, they found that when you downregulate MYD88, it also downregulates the expression. So here we're seeing a volcano plot looking at the full change of genes when you downregulate the expression of MY88 on the x-axis, and then the response of these other genes on the y-axis. And so what they found was when you repress MYD88, you also get a repression of immunoglobulin heavy chain proteins like IgHG1 and IgHG2B. Next is to see whether or not this result was specific to the first dose, or can you see the same result again after treating a mouse with the same experiment 21 days later or some time after your initial injection. And they do this because mice, or in humans too, they tend to have some immune response to the vector, the virus vector. And re-administration of the virus vector would not work because there is some immune response already built to fight off the adenovector, the virus vector. And so what they did was, for the first test, they did their standard test, but they wanted to measure the heavy chains, the immunoglobulin proteins, to test to see if there is immune response within these mice. And figure 3A demonstrates exactly that. So for the first administration, when they first administer the vector to the mouse with these Cas9 expressing mice, you get a significant reduction in the amount of immunoglobulin compared to its mock. And then when you look at re-administration, 21 days later from the initial point of injection, you still see some reduction. All right, so you get day 21. This is still a reduction. It's a slight decrease, but at day 50, you're getting that that repression. And you see that same sort of signal and in other proteins as well, like the IgG2A. Next is to see whether this is re-administration specific to IgGs, or if we could see this with other genes as well. So they tested the re-administration and they looked at data at 21 days, and what they found was when you significant reduction in the amount of RNA expression, or luminescence. Sorry, this is luminescence, and they're looking for the protein. They got significant reduction in either if it's a lax ZG or no total IgGs or serum with sorry, this is from a bacteria. Total IgGs here, IgGs again. Here, the serum Cas9 is a foreign entity that was used from streptomyces, I believe. But they also wanted to see whether they could repress this 21 days later. Also, if the repression of these certain genes can overexpress the LAC-C gene. And what they found was, yeah, when you're using this system, you could also see a response in expressing a certain gene as well. And then lastly, here is where they re-administered the gene, the vector, with targeting different genes throughout a 21-day span. And they analyzed the data at day 35, and they tested for antibodies, for cholesterols, the gene that they initially repressed, and the gene that they repressed 2 times at day 7 and day 21. And what they found was, yes, there's a reduction in the in immunoglobulin, so an immune response to the vectors, but they also found a significant reduction in serum, so this protein right here, as soon as they administer the drug. So not only does this vector show it can modulate an immune response, but it's working. You know, the Cas9 system is working within these mice. And you can see it here in figures G and H. When you're looking at the serum and when you're looking at overall cholesterol. Next is the authors wanted to test its durability. So not only was the time frame in the previous experiments within the same month, now is to test, can we repress the gene or use this system to repress the gene and see the repression still maintain some long time after our initial dose. And so that's what they tested here. So they injected the mice at day 1, and then 23 weeks later, they measured the repression of the MYD88. And you could see across the lungs, the blood, and the bone marrow that there was a significant reduction. So this system is still working, and it's very durable. Next is to see, okay, if it's durable throughout some long period that is 23 weeks, are there also additional side effects to these mice that we're not seeing? And they measured these side effects. I should have these names, but what you see is when you look at lipase levels in the blood, albumin is this major protein that's found in the blood. LDH levels, when they're recording all these indicators that tell you the quality of the mice or how healthy the mice are, you see no reduction in these other clinical markers when comparing it to the mock. However, I want to mention that when you're looking at these groups, especially here, so when you look at the first graph to the left in figure 4b, you have this p-value of 0.5, and you have this huge variation of the data. It's really funny to me because 1 point could really skew the significance or the acceptance for ALT. But when you look at lipase, right, an enzyme found in the blood, you have this one marker, one result that's really driving the non-significance. I believe if this point were to be somewhere up here, it would cause some damage to lipase levels. Not really so much as an albumin, but you see that here too in LDH. I believe that this CRISPR-Cas9 system throughout some long-term period does increase LDH levels because this 1 point isn't so convincing when you're looking at when you take a look at all these other 3 points here. And then when you look at weights, there's no difference, as the author stated, but when you take a look at just where the pink line is and where the burgundy line is, there's a slight increase in the weight in these mice over some long-term span after using the CRISPR-Cas9 technology on them. Now is to test its clinical potential, which is, can this CRISPR system, using the repressors, using the 14 nucleotide length truncated guide RNA, can this act as a prophylactic measure against some sort of infection in the blood or an infection? And they did that by injecting the vector at day 1, then injecting LPS, which is, I have the word right here. Oh, I do not have the word right here. Lipopolysaccharides. Lipopolysaccharides that should cause some sort of immune response. And that's, so by using this, they were able to test, okay, can we treat them, and then can we expose them to some sort of foreign entity, and do they have a response to these entities? And so that's what figure 4b, D, and C. Figure B and D are demonstrating here, is that when you look at those same immune response genes, MYAD88, ICAM1, and TNF alpha, you have a significant reduction when using the CRISPR system and reducing the amount of those immune response genes compared to its mock, shown in pink, comparing it to the Burgundy group. And then they measured lactate levels in the blood after using this system as a prophylactic measure, and yes, it can be used to reduce the amount of the immune response. But not only did they look at these 3 genes and lactate levels, they looked at a variety of genes that are known to have some sort of immune response or inflammatory response. And let me see, why was that there? You could look at this P map by splitting the top and splitting the bottom. The bottom is basically your control, and the top is your experiment where they repress the myad gene. And what you're able to see is that the bottom, which has this mock and it's not repressing this gene, you find a majority of these genes in red, which indicate higher expression, or they're found more in the blood. I mean, more they're being expressed more. However, when you look at the repression of MyAD88, you see the opposite, where you see less red and a little bit more blue. So this supports the idea that, yes, this system can act as a prophylactic and it modulates this immune response. So now, moving on to a more of a realistic sort of point of view, now let's test this method or this idea on wild-type mice, mice that do not express the Cas9 protein. And so what they did was they injected both the vector, the guide RNA, and the Cas9 protein within this vector. Then they exposed these same mice to the lipopolysaccharides, and then they recorded the data. What they found was the same exact thing as the same exact results as previously before. So you have a significant reduction in myad transcription, ICAM1 transcription, TNF alpha, NFC, NCF, IL-6, and IL-1 beta. You see a reduction in these genes that are related to MyAD88 across by using this delivery system. Lastly, lastly, for figure 6, to see if they could act as a prophylactic, is that they looked at lactate levels like before, and what you see is like this same exact figure, actually quite suspicious, you could say. You see the same exact response when you're looking at the transgenic mice versus its wild-type mouse. Looking at the responses of this CRISPR repression, and you get some repression in myad, and then you get the same exact repression when it's exposed to some pathogenic entity. Next is to do the opposite. So now, not see if it could act as a prophylactic, but what happens when the mouse is exposed to that pathogen or pathogenic or foreign entity, the thing that causes an inflammatory response, and then use CRISPR. And here, and you use CRISPR to modulate that immune response, to see if we could control the immune proteins, immune genes related to that inflammation. And so what they did was they injected these mice with lipopolysaccharides, waited until there was some sort of inflammation or an inflammation cue, then used CRISPR to reduce the amount of inflammation. Or if they could reduce the amount of genes that are related to inflammation. And then they test 72 hours later, they tested for the expression of those genes. And they did this through the form of a nanoparticle. So this is not a vector anymore. This is a lipid-based nanoparticle. So the same sort of delivery system that they're using for the mRNA vaccine, they're doing that for carrying the Cas9 protein, the guide RNA, the repressor proteins. What you find is this, and this is what in Figure 7 demonstrates the results of seeing if CRISPR can act as a protective mechanism against LPS inflammation. And what I want you to focus on is looking at all the pinks compared to the maroons. All of these groups are the same as usual, except you have these new proteins down here that are also related to the immune response. But overall, you take a global view of things, there's a pattern. And that pattern is that CRISPR is working and it's significantly represses that immune response across all these genes. And then next they tested for the plasma proteins. They looked at cholesterol, LDLs, HDLs. They looked at ALT, some of these biomarkers found in the blood, and they see no difference in cholesterol, a significant drop in HDL, significant drop in LDL, drop in ALT and AST when you're comparing that experiment group to the mock. So overall, the authors provide a transcriptional modality for the synthetic control of immune response in vivo. So previously, a lot of these studies have been done in vitro, where they're able to cause deletions, but they repress the gene. However, this paper says we're able to not harm the DNA, and yet still repress transcription in the gene in vivo, so in an actual living system. They're also able to look at not only one gene, but everything related to that gene, everything related to inflammation response. And so by regulating but overall, I think this paper really does, this paper demonstrates that by using by deactivating or taking out the nucleus domain on the Cas9 protein, there's a range of things you could do with the CRISPR-Cas9, and it's readily programmable. So overall, I believe that the authors did an excellent job demonstrating the range in utilizing CRISPR by showing both the efficacy in vivo and in vitro in regulating gene expression. However, one gray area, and I feel like should be revisited, and it's the long-term side effects of using CRISPR in the mouse, because as you can see here, there are no differences when you look at all these comparisons. However, when you take a deeper look at the data, there's either 1 or 2 points that really skew the significant difference in these groups. Together, that wraps up my presentation. Hopefully this all recorded and I could turn this in right after this. Thank you.